All right, so yesterday we kind of introduced the topic of cellular respiration. Today we are going to expand on it even more, going a little more in depth with cellular respiration and looking at it, the process of glycolysis, which is the first step of it. So I kind of mentioned and touched on this yesterday about how glycolysis is in the cytoplasm, and we mentioned like you start with the ball on the top of the hill and the ball's just going to sit there indefinitely until you give it a little push. Well, that's kind of what's going on with glycolysis. Is you can't really start this whole process and unless you invest a little energy to get it going. The bonds aren't just going to naturally break unless you give them that little shove to get them to break and invest a little bit of energy at the start of it. So glycolysis kind of starts this whole thing going. This is where sugar is originally going to break down. You're going to convert the 6-carbon molecule. Now, C6H12O6. We've got 6 carbons here. And we're going to kind of cut it in half, and we're going to make 2 3-carbon sugars, and these are called pyruvates. So the pyruvates are going to go into kind of the next step. So we're taking the 6-carbon glucose, and we're going to turn them into pyruvates that are 3 carbons. And this all is going to take place in the cytoplasm. So we're going from 6-carbon to 3-carbon pyruvates. And then we're also going to make a few ATP during this process.
Alright, so I want to see that.
What the sigma?
I did see what you said. What's going on my skateboard bag? Say 1 to something. I literally shift. Because I work till 5. Well, let me think. I don't want to like.
It's fine if you don't. I can't work it.
Yeah, honestly, I don't know, because I heard you should go in early on this Friday. I don't know, but I'll think.
All right, so here's our glucose. We know that glucose is in a ring. We already covered that when we learned about these macromolecules. So glucose is in a ring. It goes through the process of glycolysis, and then we have 2 pyruvates that end up from it. So it splits it kind of in half. So here's 3 carbons, here's 3 carbons. So we're splitting that glucose in half. The black ones represent carbon. So when you make these, there's like little molecule kits you can get. The black represent carbon, the red represent oxygen, and then the white represent hydrogen. So when we have this glycolysis process going on, energy does need to be used in order for this to work. So 2 ATP is invested into this. And I know I mentioned it yesterday. It's like me telling you, okay, I've got this brilliant idea. I'm going to make $4, but in order to make $4, I really need you to give me $2 first. I know that if you give me $2, I can make $4 out of it, but first you got to give me $2. I mean, it sounds like a great deal. I mean, it's like, hey, I'm going to double your money. That's basically what glycolysis is doing. It's taking 2 ATP and it's turning it into 4 ATP. So it's a total of 2 ATP needs to be spent, and you don't need to know this whole PGAL and glucose 6-phosphate. You don't need to know all that. All I want you to understand is that glucose is going in, and 2 ATP is going in, and this is taking place in the cytoplasm. I don't care about the glucose 6-phosphate and PGAL. That's a lot. I mean, phosphoglyceraldehyde is a fun word though. It's kind of fun to say. But you don't need to know that. Sometimes I think scientists just like to put really big words together just to see how long they can get them. I still think one of my favorites is sphygmomanometer.
Huh?
Sphygmomanometer. That one just kind of rolls off your tongue. That is a blood pressure cuff. The name for a blood pressure cuff is sphygmomanometer. That's why they don't ever call it that. They call it blood pressure. Or they just say it's a cuff. Make fun of us old people. Alright, now also in this process you're going to get some NADH. Remember in plants we had NADPH. So you can remember that in plants it was P for plants. But here we have NADH. Now the NADH is important. We're going to need this again. Nothing is wasted in these processes. Everything has a purpose. So the NADH is going to be used again. We'll see that come back in the electron transport chain. But we do make some NADH during glycolysis, and that gets saved and it gets used again. So again, I don't care about the PGAs and the PGALs and all of that. I do want you to know that NADH is made and that's going to be used again.
They didn't. I picked that time. A larvar in his foreign regions. It's kind of okay. Is dates, peanuts, chocolate chips, and sea salt. Sounds too healthy for me. It's steaming.
Healthy option, and then she goes, Dorito. And then she goes, yes please, Dorito. Oh, it's so funny. Oh, you guys are hilarious. Riley, do you get along with your brother?
Yeah.
Yeah.
Why?
I'm just curious, because some siblings don't get along, and you and your brother, you strike me as like you get along pretty well. Yeah. I have 4 siblings. Is it just the 2 of you? Do you have any other siblings?
Or just music.
I have 4 siblings, and we all get along really well. We're pretty close. And my children, I think my children are pretty close, and I love seeing that in them. So. But yeah, you and Blake seem like you get along pretty well.
Me and Jackie are really close, but we definitely argue a lot.
Well,
Me and Blake argue a lot too.
siblings argue.
We also share a room, though.
Yeah.
I hate my brothers.
Me and my brother got along once he got a girlfriend. He became much nicer. And like he would let me go with him everywhere and take me to the mall.
My brother is dating my cousin.
Yeah. But and he's nicer now that I don't see him that much too. Well, like he's nicer, not nicer that way, but he's nicer.
We fought growing up, my siblings and I, but even like fighting, we were still really close.
My sister annoys me, and that's another thing.
There's also a huge age gap.
There is age.
We were all fairly close in age too, and my siblings or my children are pretty close in age.
Me and my brother are 15 years.
That's good.
Me and my brother are 3.
Everybody
Me
is-
and my sister are
Well,
10.
there's 2 years in between each kid in my family besides me and twins. What's your 7?
It's not good.
There's 2 years between her.
How many siblings do you have?
There's one younger. scratch my
Why would they have the same thing?
Well, she just said, she said,
It's easier to say years and 8 years and 18 months. Yeah.
well, she said that I'm not sure.
I think having siblings
She
is
said
fun.
6 months versus.
I'm glad I grew up with siblings, and I'm glad I was able to give my kids siblings.
Because 6 months is a year.
Alright, anyways. So, steps of glycolysis, 2 ATPs form, energy is created, and we know 2 ATP were put into it. So you create 2, and then there's something called substrate level phosphorylation. This is the direct transfer of a phosphate group from ADP. And so glycolysis ends with the formation of 2 more ATP. So we have 4 ATP created. So it will ask you on the test how many ATP are formed in glycolysis. A good way to answer that would be 4 are created, but only 2 of them really count toward the overall total. Do you understand why?
Because 2 are being.
Yes. Good. So 4 ATP are created in glycolysis, but only 2 of them count toward the overall total of ATP because we had to spend 2 to get the process started. Because you will definitely have a question about how many ATP are created in cellular respiration and at which steps are the ATP created. So you get 4 during glycolysis, but only 2 of those really count. You also get NADH here. What goes in, what comes out, where it takes place. These are the things I'm looking for.
My hands have been really cold recently.
My hands always get dry.
So do you guys have gloves at your house?
Yeah.
Okay, so I might be worried I forgot to pack my gloves. I think I did though.
This is a great summary slide. 2 ATP per molecule of glucose. A total of 84 are formed, but only 2 were to count because 2 were invested in it. Glycolysis also produces 2 pyruvates. Also should have put 2 NADHs up there as well. But 2 pyruvates also come out of this process. So just a really good overall summary slide.
Should I go refocus, please? Thank you.
Whoa. Why? What the heck? What?
Alright, so again, just another summary slide. Glycolysis occurs in the cytoplasm, breaks apart 1 glucose, uses ATP, yields 4, but only 2 can count. You get 2 pyruvates, and you get NADH. So that's the whole glycolysis process. And we're gonna watch a little video about it.
Miss Harris, do you know how soon our test will be?
Probably before Thanksgiving. That's what I'm kind of aiming for. Well, so, yeah.
Glycolysis is the breakdown of glucose into two molecules of pyruvate. It begins with an energy input of two ATP molecules. First, enzyme action promotes the transfer of a phosphate group from ATP to glucose to form glucose 6-phosphate. The glucose 6-phosphate molecule rearranges to form fructose 6-phosphate. A second phosphate is transferred from another ATP to form fructose 1,6-bisphosphate. This molecule immediately splits to form two molecules of phosphoglutaraldehyde, or PGAL. Each PGAL molecule gives up 2 electrons and a hydrogen to NAD to form NADH. Then each PGAL combines with inorganic phosphate to form a molecule of 1,3-bisphosphoglycerate. Each molecule of bisphosphoglycerate donates a phosphate group to ADP to form ATP. With this formation of 2 ATPs, the original ATP debt is paid off. Each of the resulting 3 phosphoglycerates undergoes a rearrangement and then releases a proton and 1 hydroxide ion, which combine to form water. The resulting intermediates, 2 molecules of phosphoenolpyruvate, or PEP, are unstable. Each gives up a phosphate to ADP. Once again, 2 ATPs are formed by substrate-level phosphorylation. The end products of glycolysis are 2 molecules of pyruvate.
So again, you don't need to know all the, like, PEP and all that stuff. That's not what I am concerned you understand. Okay. So this shows the process step by step, what's going on. Here's where we get some ATP going in. Here's where we get some ATP coming out. Okay. Now we've got some pyruvates. That's great. But the pyruvates aren't super helpful. We've got to change it up a bit. We can't really use the pyruvates for the next step. So we have to do something called the acetyl-CoA formation. We've got to take those pyruvates and we have to modify them a little bit. So we've got to change them in order to put them into the Krebs cycle. So that's what the acetyl-CoA formation does. It's kind of like an in-between step. We're getting them ready to go into this Krebs cycle or the citric acid cycle. And so we take the pyruvates, we modify them a little bit, and then we put them into this Krebs cycle. So that's what the acetyl-CoA formation does. We're changing the pyruvates to get them ready for the next step. This does occur in the mitochondria. In fact, all of the next steps are going to occur in the mitochondria. But remember, since we have 2 pyruvates now, all of these reactions from here on out are occurring twice, 1 for each pyruvate.
And the Krebs cycle was on my diet.
It should be.
It is. It is.
I understand a whole lot of words.
Same.
I didn't even realize. I want the brownies.
Remember when I did that when I was still my driving and I was saying Are you recording?
Yes.
She's recording. No. Just beep it out. Just edit it.
You're gonna have to censor that out.
Anyways,
Moving on. Back to biology.
back to biology. So we have our two pyruvates. They go into the acetyl-CoA formation. And then here we have a little bit of a breakdown. With the breakdown, we have some CO2 coming out, and we have some NADH coming out. So more NADH. Because we've changed it from a 3-carbon into a 2-carbon, that's where some carbon dioxide is coming. And this is one place where carbon dioxide comes out. And now we have the acetyl-CoA going into the citric acid cycle. So we lose a little bit of carbon dioxide here. We get a little bit of NADH. This is going to go into another process later on. So remember, nothing wasted. Everything has purpose. These things are going to go, well, carbon dioxide goes into photosynthesis. NADH is going to go into the electron transport chain. And the acetyl-CoA is going to go into the Krebs cycle, or citric acid cycle. So all of this has a purpose, and we'll see this later. So the Krebs cycle, we're going to get some ATP, some FADH, and some NADH. These are all going to go, the FADH and NADH is going to go to the electron transport chain also. So again, we went from the cytoplasm into the mitochondria. Here are the pyruvates. The pyruvates are going into our acetyl-CoA formation. That's going into our Krebs cycle. And from there, we are going into the electron transport chain, which will happen next. But the Krebs cycle is going to give us some carbon dioxide, some ATP, NADH, and FADH. All right, so again, I want you to know what goes in, what comes out, and where it takes place, whether it's in the cytoplasm or the mitochondria. So here's the acetyl-CoA formation, what's going on. It's in the mitochondria, and now you've got the pyruvate. Instead of a 3-carbon, which is what goes in, we're having a 2-carbon, which is an acetyl group. So we split the 3-carbon into a 2-carbon. Here's our 2-carbon. And one of the carbon dioxides is going to go away. That's going to go out of the cell, or we're breathing it out in our case. And eventually it's going to be picked up for photosynthesis. We're also going to get some NADH. This is going to go to the electron transport chain. We'll see this again. We'll see NADH again.
Matrix.
The matrix is, you know how we had the stroma in the chloroplast?
Yeah.
Is the fluid part? Think of the matrix as the stroma.
Okay.
And also cytoplasm. It's the fluidy part inside the mitochondria.
Yeah, I was thinking like Matrix, like the Matrix. Yeah.
No. It's the fluidy part inside the mitochondria. Just like the stroma was the fluidy part inside the chloroplast, just like the cytoplasm is the fluidy part inside the cell. They just want to name them all different. You just can't pick a name and stick with it.
Pick a name.
You would think, no.
Or make the names like really, really similar.
And then nucleoplasm. Now that at
Or
least
make
it's
it-
kind of similar.
They make it to where it's the opposite of what it means.
Yeah.
They just like the trigger.
Like photosystem 1 and photosystem 2. Backwards. But that one I can kind of get because photosystem 1 came first. That one I can kind of get.
But it's second.
Yeah.
Hurry up.
I know, she didn't realize. Oh, that's the bowl. I need to. I don't know why my phone is like you're bad at it.
That was like what I was talking about. Like I was thinking of this, like I was driving in a car with like.
Does everybody get this one?
1 second here. Don't let me know. Okay, I got it.
Good. So the steps of the Krebs cycle, it's very similar to what we saw in the Calvin-Benson cycle. You're gonna have some energy that's invested into it, but then it's going to need to reset itself, so it's going to need a little energy to reset itself. You're going to have 2 carbon dioxides form, and then it's going to leave the cell because you're breaking apart the rest of that, what was left of that glucose. So all the glucose and all the bonds are getting broken apart. And the NADs are going to form NADHs, and that's going to be used again later as well. So everything, the rest of that sugar is going to be gone. And you're making 2 more ATPs from this process as well. It's all taking part in the fluidy part of the mitochondria, which is called the mitochondrial matrix. We're not doing anything with the membrane of the mitochondria yet. That'll come in a little bit.
What? Stop sitting in a syringe.
We good with this?
Yeah.
Okay. Oops, sorry, I went too fast. So we also have some ATPs formed in the citric acid cycle. And then we also have what are called FADs, which are going to be made into FADHs. Now, we learned about coenzymes a little bit. FADHs, NADHs, these are, think of them as alternative sources of energy. They're just not as good as ATPs. ATP is the best energy our body can get. It's really, really efficient. FADHs and NADHs are types of energies as well. They're just not quite as good. Our body can't use them as well. So if our body can think of a way to use them a little bit to make more ATP, that's what it's going to do. So it's going to use the FADH and the NADH kind of differently to make more ATP. That's its purpose.
I was so mad at him. Well, good thing I took the one. Yeah. So, you were.
So by the time the citric acid cycle is done, all 6 carbon dioxides have been formed, so all the carbon is gone from our original glucose. There's no more carbon. The citric acid cycle has also made 2 more ATP, so by the time we've done glucose and the citric acid cycle, we've got 4 total ATP, 6 if we're counting the other 2, but 4 new ATP. And we've got 8 NADHs and 2 FADHs. So we've got some alternative energy that we can use for our next step, which we will be using for our next step. I believe we'll learn the step on Tuesday. I think we're almost done. So to sum it up, uses 2 pyruvates from glycolysis, happens in the mitochondrial, the matrix to be precise, and carbon dioxide and NADH are produced, also some FADH are produced, and the 6 carbon dioxides are released, and 2 ATP are formed. And we'll watch a video.
The second stage reactions occur in a mitochondrion's inner compartment. In the first preparatory reaction, a carbon atom is stripped from pyruvate and released as carbon dioxide. The remaining carbons combine with coenzyme A and give up hydrogen and electrons to NAD. The product of these reactions is acetyl-CoA. The acetyl-CoA will enter the Krebs cycle. In the first steps of the cycle, acetyl-CoA transfers 2 carbons to oxaloacetate to form citrate. Next, another carbon atom is released as carbon dioxide, and NADH forms as NAD picks up hydrogen and electrons. Another carbon dioxide is released, and another NADH forms. At this point, 3 carbon atoms have been released as carbon dioxide. This balances out the 3 carbon atoms that entered the mitochondrion as pyruvate. 1 ATP forms by substrate-level phosphorylation. FADH2 forms when FAD picks up electrons and hydrogen. The final steps regenerate oxaloacetate and form 1 more NADH. For each molecule of pyruvate, the second stage reactions produce 1 ATP and reduce 4 NAD to NADH and 1 FAD to FADH.
Okay, this just shows the cycle. We've got NADH coming out, FADH, ATP being produced, and then, oh, actually it's going this way. It's going that way. And then the cycle does have to reset itself. So we are going to have to add some ATP to reset the cycle so that it can repeat itself. And then we have the 2 pyruvates coming in, each with a 3 carbon. And then by the time we're done, we have 6 carbons coming out. All right, we're done. Fermentation lab. So this week we have the photosynthesis lab due, and we have the cell drawings due, the cellular respiration photosynthesis drawings due. Sound good?
Yep.
Okay, all right, get those done. Fermentation lab due next week.